Today, we're going to be unearthing the mysteries of ancient deep-sea ecosystems. What was life like in the darkest depths? How did those strange and wonderful animals get their food? And how did they survive when there was no light to sustain life? Spoiler alert, it's weirder than you think. So let's dive into it. So what do we actually mean by deep sea? A commonly cited definition is any area below the photic zone where sunlight can penetrate and support photosynthesis. This depth varies with latitude and water clarity, but it's generally accepted to be around 200 meters or about 650 feet. In this zone, we find creatures adapted for extremely high pressures, freezing temperatures, and perpetual darkness. Some animals, like the Japanese anglerfish, have bioluminescent lures to attract prey in the darkness. Others, like the tripod fish, have downward-facing eyes to spot prey against the faint sunlit surface above. But how does the fossil record help us understand these unique environments? Well, fossils are the preserved remains or traces of ancient organisms. They provide direct evidence of past life and are the primary tool for reconstructing ancient ecosystems. Unfortunately, the fossil record of deep-sea life is relatively sparse compared to shallower environments. This is mainly due to the challenges of fossilization itself. As mentioned in our previous episode, fossilization requires a combination of factors that are more likely to occur on land or in shallow waters. The conditions that typically lead to fossilization, such as rapid burial and the presence of hard parts like shells or skeletons, are less common in the deep sea. The deep sea environment is also characterized by high levels of pressure and low oxygen, which can make fossilization even more unlikely. Additionally, the remote location of deep-sea habitats makes them less accessible for both living organisms and the geological processes that can lead to fossilization. However, there have been notable discoveries of well-preserved deep-sea fossils in places like the Peru-Chile Trench and the Japan Sea, thanks to special circumstances like extremely slow sedimentation rates, cold oxygen-rich bottom waters, and the absence of scavengers. These rare examples provide invaluable insights into ancient deep-sea ecosystems. But another challenge is that deep-sea animals tend to be soft-bodied. You might be thinking of those bizarre blobfish who look like something between a shapeless blob and a smiley face. Their lack of distinct hard parts makes fossilization even more unlikely. But paleontologists have found some solutions to this problem. One example is the discovery of a fossilized poop containing undigested fish remains found in the Kerguelen Plateau in the Indian Ocean. This coprolite provided evidence of a new species of anglerfish from about 70 million years ago and helped researchers better understand the diet of this mysterious creature. Another example is the discovery of jellyfish fossils in the Black Sea. Jellyfish have soft, squishy bodies without any hard parts at all. However, the unique conditions in the Black Sea have allowed for their preservation in a form called permineralization. In this process, minerals seep into the tissues of an organism after it dies, creating a petrified replica. These jellyfish fossils have helped scientists learn about the evolution and distribution of these animals over time. The scarcity of deep-sea fossils presents a significant challenge for understanding ancient deep-sea ecosystems. The lack of representative fossils makes it difficult to study the diversity, abundance, and interactions of organisms in these environments. It also makes it challenging to reconstruct the evolutionary history of deep-sea life and to understand how ancient deep-sea ecosystems functioned. To address this challenge, scientists are exploring new techniques and approaches. For instance, they are investigating alternative methods for discovering and preserving fossils, such as studying microfossils or trace fossils. Microfossils are tiny fossils that are often overlooked, while trace fossils are the preserved evidence of an animal's activity, such as footprints or burrows. By studying these types of fossils, scientists can gain a better understanding of the diversity of life in ancient deep-sea ecosystems, even in the absence of traditional body fossils. Moreover, scientists are utilizing advanced technologies to analyze existing fossils in new ways. For example, they are using high-resolution imaging techniques to study the morphology of delicate deep-sea fossils in great detail. These studies have revealed new insights into the adaptations of deep-sea animals to their environment, such as the development of specialized sensory systems for detecting prey or navigating in the dark. Despite the challenges posed by the limited fossil record, scientists are continually making new discoveries about ancient deep-sea ecosystems that are shedding light on the history of life on Earth. Let's talk about one particular discovery that helps shed light on the mysterious deep-sea food web. Researchers in Japan studied rocks formed from underwater volcanic ash that were millions of years old. They discovered a bloom of a type of radiolarian, a single-celled marine organism with a silica shell. This event took place approximately 52 million years ago and was likely triggered by a massive eruption of Mount Ibu in Indonesia. While not directly related to the deep-sea food web, the eruption would have had far-reaching effects on ocean chemistry. This suggests that changes in nutrient availability could influence the distribution and abundance of different species in the ecosystem. Specifically, the researchers suggest that a decrease in nutrients in the surface water may have caused a collapse in the population of euphausiids, also known as krill. This ecological disaster cascaded down the food chain, affecting other animals like fish and whales. The team used a variety of methods to study these ancient creatures. For instance, they analyzed the chemical composition of individual molecules from single cells, revealing the types of lipids present in each organism. This analysis showed that the diet of different species varied widely. Some, like foraminifera, relied primarily on marine snow for sustenance, while others, like radiolarians, consumed phytoplankton. The radiolarians studied were heterotrophic, meaning they obtained nutrients by consuming organic matter. Their diet consisted of marine snow, fecal pellets from other organisms, and particles from the decomposition of dead animals. Marine snow is a significant source of food in the deep sea, consisting of a mix of organic and inorganic matter that falls from the surface waters. By studying the carbon and nitrogen isotopes in the radiolarians' shells, researchers could reconstruct the food web dynamics of the time. The data indicated that the radiolarians consumed a mix of fresh and decomposed matter, but the majority of their diet consisted of marine snow. The team also used lipid biomarkers to study the diets of foraminifera, another group of single-celled organisms found in marine sediments. The fatty acids and sterols present in the cell membranes of different organisms serve as indicators of their dietary preferences. Foraminifera that consumed phytoplankton had higher levels of polyunsaturated fatty acids, while those that ate bacteria had higher levels of branched and cyclic isoprenoids. The findings highlight the diversity of feeding strategies employed by single-celled organisms in ancient deep-sea ecosystems and provide insights into the complex interactions within the food web. Overall, these studies contribute to a better understanding of the ecology and evolution of deep-sea life during the early stages of the modern ocean's development. Let's look at one last example. Scientists recently discovered fossils of deep-sea creatures from about 180 million years ago off the coast of New Zealand. The fossils include sponges, jellyfish, and the oldest known fossils of deep-sea corals in the Tasman Sea. These discoveries provide insights into the biodiversity and environmental conditions of the deep sea during the age of dinosaurs. Prior to these discoveries, little was known about the deep-sea ecosystems in this region during the Mesozoic era. The fossils of sponges and jellyfish indicate the presence of a diverse range of marine organisms in the deep waters of New Zealand during the Jurassic period. Sponges are filter feeders that rely on currents to bring food to them. Their presence suggests that the water column was likely well mixed, allowing for the transport of nutrients and oxygen to the deep sea. Jellyfish, on the other hand, are mobile predators that play an important role in the deep sea food web. Their fossils indicate the presence of a thriving community of fish and other prey animals in the area. The discovery of deep-sea corals is particularly significant as corals are sensitive to environmental changes. Their ability to thrive in the deep sea during the Jurassic period suggests that the water temperature and oxygen levels were suitable for their growth. Additionally, the presence of coral reefs may have provided habitat and food sources for other deep-sea organisms. Overall, these fossils paint a picture of a dynamic and diverse deep-sea ecosystem that supported a wide range of marine life during the age of dinosaurs. Well, that's a wrap on today's episode. We hope you learned something new and exciting about the ancient deep sea. If so, please don't forget to leave us a like. And if you love what we do, consider supporting us to keep these fun science adventures coming. Thanks for joining us, and we'll see you next time.